Hello, this is Dr. Morris, and what we're going to be doing in this brief lecture, relatively brief lecture, is reviewing some concepts from undergraduate physiology, and specifically some of the concepts in cellular physiology and a description of cell division that is just meant to help you in terms of your understanding as we get into clinical physiology and the basis for how we are going to be understanding health and disease using physiology. You all remember that a cell is the smallest unit of life within an organism, that the cell consists of its internal contents, cytoplasm and organelles, that are bound on the exterior surface by a nuclear membrane. So we have intercellular fluid, which is the cytoplasm, and we have interstitial fluid, which is also called extracellular fluid. Extracellular fluid or interstitial fluid is the fluid that is outside of cells throughout the body. Interstitial fluid really is describing the fluid, the solution that is between and around cells. So the cytoplasm, or the fluid inside the cell, the intracellular fluid, is separated from the extracellular interstitial fluid by a cellular membrane. And we'll talk a little bit about how the cellular membrane is constructed in a minute. But I want to start by talking about fluid. 75 to 85% of most cells, not all cells, but most cells, is composed of water. Water within which are solutes, and we'll talk about the solutes because an awful lot of life processes are dependent upon those solutes. 60 to 80% of body weight is water, and that is certainly dependent on some other variables. So people become less water with age, obesity, and perhaps muscle mass. All those things factor into the percentage of body weight that is composed of water. Okay, so water in which things are dissolved. And one of the important sets of solutes are salts. And these include things like sodium chloride or just salt, along with potassium salts, magnesium salts, calcium salts, which are not on this slide. And those salts can be chlorides, phosphates, sulfates. So we see that along with the cations, we have anions, and that the anions include chloride, phosphate, and sulfate. Now, one of the things I want you to notice on this slide is just a diagram, but it's illustrating that in the intracellular fluid, the concentration of certain ions is higher than it is in the extracellular fluid. And the things to pay most attention to are that potassium exists at a higher concentration within the cell compared to sodium, which is in a higher concentration outside of the cell. There is potassium outside of the cell. There is sodium inside of the cell. But this differential between the concentrations of potassium and sodium within and without the cell is an extremely important one. So I want you to remember, outside of the cell, more sodium; inside of the cell, more potassium. Much of the chloride stays outside of the cell, and the important anions inside of the cell tend to be things like phosphates and sulfates. So remember that. And in fact, as we go through this course, I'm going to be asking you to remember some numbers. Right now, we won't do that, but starting at our first lecture, we will. And those numbers are important because these are laboratory values that you're going to be seeing every day of your professional life. we can measure the ions in extracellular fluid. We do that by taking a blood sample, spinning it to take off the cells so that we're only getting extracellular concentrations. Plasma are actually serum concentrations, and we measure them. We can't measure ions intracellularly, and in fact, even when we're measuring extracellular ions, it's really difficult to know based on those values, what's going on in terms of the whole body with deficits, with excesses or deficits of those ions. So just because, for instance, potassium is low in the serum does not mean that total body potassium is low. Perhaps something has happened that has driven the potassium into the cells, say high levels of insulin, or certain drugs, in which case that perhaps we have an excess of potassium inside of the cells and a deficit outside of the cells and a normal total body amount of deficit. So just be aware of that, because it makes us have to be careful when we're replacing ions, which we often do in IV fluids. There's just no good way to calculate deficits. So along with water and along with these solutes that are our ions, our salts, we also have protein in the cell, and proteins make up about 10 to 20% of cell mass. There's actually usually more protein outside of the cell, but the proteins within the cells consist of structural proteins, and those include proteins that make up the actual structure of the cell and things like microtubules, and as we go further into cell structure and organelles, that will be helpful. In addition, there are globular proteins, and many of these are enzymes, some of them produced for use within the cell, some of them produced to be extruded from the cell and used by the body in another way. So just be aware that protein is part of the construction of the cell. There are lipids in cells. Now, most cells, those lipids consist primarily of phospholipids and cholesterol. And as we'll see in a couple of minutes, those are used to form membranes, and there are many membranes within cells. Triglycerides are another class of lipids that are important. And they exist in large amounts in the cell type called adipocytes, fat cells. And these are pictures of adipocytes where you can see that the cytosol is really virtually entire fat, and entirely fat. The nucleus is pushed up against the side of the storage cell, against the cell membrane. And in adipocytes, triglycerides are about 95% of the cell mass. There are also carbohydrates. So when we're talking about these major classes of compounds, proteins, fats, and carbohydrates. In most cells, there's a small amount of carbohydrate. It's often glucose, and it's continuously being brought into the cell and used in the process that you know is the Krebs cycle to make energy for the cell and ultimately to convert ADP to ATP. So there's only a little bit of carbohydrate, and it's constantly being moved. But in certain cells, muscle cells for instance, and especially hepatocytes, cellular carbohydrate consists as glycogen, which is a polymer of glucose that is important for storage to release glucose when it's needed. In addition, there is carbohydrate in many cases attached to proteins. Those are called glycoproteins, and we'll come to that in a minute. The physical structure of the cell, as I said, was that we have the cellular membrane. Within the cellular membrane is the cytosol. Within the cytosol are a number of physical structures that we call organelles, and we'll talk about what many of the organelles do. Most of the organelles are also surrounded by membranes. Those membranes, as I said earlier, composed of lipids, specifically phospholipid and cholesterol, along with structures that are made of proteins that perform a number of tasks within those membranes. So when we're talking about membranes, we have the cell membrane itself, the outer membrane, the nuclear membrane surrounding the nucleus, and contiguous with the membranes of some of the other organelles, particularly of the endoplasmic reticulum, and we'll come back to that as we look at diagrams and talk about the structure and function of some of these organelles. But they are all surrounded by lipid membranes. The cell membrane itself is a lipid bilayer. So we'll show you that in a minute. It's a lipid bilayer composed of phospholipids and cholesterol. And as with many fats at body temperature, they are liquid and they flow. So a cell membrane isn't a solid, it's a liquid. And the proteins and other substances that are either dissolved or suspended in that bilayer can move. They diffuse so that they're existing in all areas of the cell membrane. Okay. Those phospholipids have a hydrophobic end that rejects water and a hydrophilic end that will interact with water. And the reason that this is a bilayer is that those hydrophobic ends end up pointing in, facing each other, and the hydrophilic portions face out to interact with both the interstitial fluid and the intracellular fluid. And here we have an electron micrograph of actually two cell membranes. So this is cytosol, this is cytosol. Here's actually an organelle called a lysosome. You can see that this membrane is composed of two layers of lipids. And what you can't see here very well are the proteins that are suspended and floating and dissolved within that membrane. You can see one membrane, then interstitial fluid in the intercellular space, and then another membrane, and then the cytosol of that cell. So because these membranes are lipids, they're very permeable to lipid-soluble substances. Cholesterol and many hormones that are cholesterol-based, steroid hormones. So corticosteroids you've heard of, things like prednisone. The sex hormones, testosterone, estrogen, progesterone, are all fat-soluble. And in addition, oxygen and carbon dioxide can permeate through cell membranes. So can alcohol and other oil-soluble solvents. On the other hand, water and those charged ions within the water do not cross cell membranes easily, and so there have to be other structures that allow those substances to pass, and those are pumps and pores. We'll talk about them in the first classroom lecture. Cholesterol controls the permeability and the fluidity of the cell membrane. This is simply a diagram showing the phospholipids here with their hydrophobic portions facing in and their hydrophilic portions facing out. And then within this membrane, and they don't show here that there is also a good deal of cholesterol dissolved in this and circulating, but there are proteins and glycoproteins, and some of those proteins go from the cytosol through to the exterior surface, to the outside of the cell, so inside to the outside cell. They're called transmembrane proteins. Some of those transmembrane proteins have carbohydrates that are attached to them, making them glycoproteins. There are also proteins that exist only on the inside or the outside. They're called peripheral proteins. They stay on one side of the membrane. Many of these proteins function as things like attachment sites for substances such as hormones, so they are receptors. They can also be parts of pumps and pores. Some of the carbohydrates in cell membranes. So I mentioned carbohydrates in cells being glucose that is coming into the cell and being used, glycogen that is being stored, but in addition there are these carbohydrate entities that are attached to proteins and to lipids, forming glycoproteins and glycolipids. Most often the glycol portion of those membranes protrudes to the outsides of the cells. Carbohydrates in these proteoglycans are negatively charged. They repel other negatively charged membranes, which moves them apart as they diffuse around on the cell membranes. The glycocalyx allows cells to adhere to each other. Some, as I mentioned, act as receptors for binding hormones. Some enter into immune reactions in that they function as antigens on a cell surface. If you think about the A and B antigens that exist on red cell surfaces, they are carbohydrates, and they are very important in the immune reaction when a transfusion is given. We'll talk about that in a couple of months. So the cytosol has water and dissolved proteins and electrolytes and glucose. There are also little globules of fat, granules of glycogen, and then things like ribosomes, which we'll talk about, which can exist either floating freely in the cytoplasm, more often are attached to the endoplasmic reticulum, and vesicles that have been formed by the organelles and are floating in the cytosol, perhaps moving peripherally in order to secrete those substances, to merge with the cell membrane and secrete those substances. So let's first talk about the endoplasmic reticulum. The endoplasmic reticulum is a really important part of cell function and structure. It's a network of tubules that penetrate much of the cytoplasm. And on the surface of the endoplasmic reticulum, which is a membrane, and that membrane is formed, as is the cellular membrane, of lipids, phospholipids and cholesterol, there are many substances synthesized. So proteins, carbohydrates, lipids, sometimes structures. So once a substance, such as, for instance, a proteinaceous enzyme is produced by the endoplasmic reticulum, it can be pinched off into a little, what's called a vesicle, which forms a lysosome or a peroxisome or a secretory vesicle. And these then are within the cytoplasm, separated from endoplasmic reticulum. Lipids are made in the smooth endoplasmic reticulum within the wall. So the endoplasmic reticulum synthesizes the enzymes that break down glycogen, storage carbohydrate, so it can be used. They also function as the site of a number of reactions which detoxify things that are not good for the cell, that might damage the cell. drugs, toxins, and there are a number of processes that are used, and we'll talk about these a good deal when we get to the gastrointestinal portion of our course, because the gastrointestinal tract, particularly the liver, functions as a major detoxifying organ, and the liver cells, within the liver cells, the ER is doing these things like oxidizing, hydrolyzing, conjugating substances in order to detoxify them. So here's a pretty diagram of the endoplasmic reticulum. One of the things that you need to be aware of is that endoplasmic reticulum may be studded with ribosomes, which are the organs that actually, or the organelles that actually produce proteins, and that is called rough endoplasmic reticulum. When I talked earlier about how vesicles can be pinched off containing proteins such as enzymes, here we have vesicles forming and being pinched off. Those vesicles might stay within the cytoplasm, or they might, considering that they're lipids, they might actually merge with the cell membrane, releasing those enzymes into the interstitial fluid in some cases. Something that is not terribly apparent here, but that we'll come back to and I'll show you in a minute, is that the membrane of the endoplasmic reticulum is continuous with the membrane of the nucleus. Okay. So within the rough endoplasmic reticulum, the ribosomes make proteins. Ribosomes are composed largely of RNA, and there is transfer RNA and messenger RNA. We'll talk about those a little more in a couple minutes. But what happens to some of those proteins is that they are transferred from the endoplasmic reticulum to the Golgi apparatus, within which they are modified. Sometimes they are cut in certain places. They may be folded. Folding is really important to the function of proteins. As you know, when a protein is first made by a ribosome, it is simply a long strand of amino acids, and those amino acids, unless they assume the right shape, are not going to be performing their function as a protein. So the Golgi apparatus looks similar in some ways to the endoplasmic reticulum. It has similar membranes. It has a bi-layer lipid membrane that's similar to smooth endoplasmic reticulum. You see a lot of the Golgi apparatus in cells that are secreting substances. So these might be cells that are creating hormones. For instance, thyroid cells, or cells within the pituitary gland. And they are, while the endoplasmic reticulum is perinuclear, it's located next to the nucleus, and as I said, continuous with the nucleus, the Golgi apparatus tend to be located on the side of the cell from which substances are extruded. Another kind of cell that is secreting might be cells within the gastrointestinal tract that are making enzymes to help us digest our food. So vesicles pinch off from the ER, as I showed you in that diagram, and then they fuse with the Golgi apparatus. Those vesicles are called transport vesicles, and they transport substances that are synthesized in the ER to the Golgi apparatus to be modified and changed. So, as I've said before, lysosomes are vesicles. They stay within the cell. Lysosomes tend to contain enzymes which are important for processing and destroying things like bacteria, perhaps, that get inside, or phagocytosed by a white blood cell. or sometimes actually for tearing down, breaking down substances within the cell as part of the cell's life cycle. Secretory vesicles are vesicles that are going to go merge with the cell membrane and then release the substances within those vesicles to the interstitial fluid, and there are other things made by the Golgi apparatus or processed by the Golgi apparatus. So here you can see an electron micrograph and a diagram, again showing how this is similar in some ways to the smooth or very similar in appearance to the smooth endoplasmic reticulum. And again, we have these vesicles, these transport vesicles coming in and merging with the Golgi apparatus from the endoplasmic reticulum, and then we have vesicles budding off and either being lysosomes within the cell fluid, the cytosol, or being transport vesicles carrying secretory vesicles carrying enzymes to the cell wall. I think we've said this already. I actually went backwards, sorry. So, lysosomes contain digestive enzymes. They're the digestive system of a cell. And they break down damaged structures. They break down food particles, things like glycogen granules and bacteria. If a lysosome is damaged, if it's breached, it might be to perform an important function like breaking down a damaged area, but it might also damage the cell if those enzymes are not meant to be breaking down some specific substance. Moving on to our next type of organelle, the mitochondria. So the mitochondria are extremely important organelles. They contain their own DNA, and they reproduce, they replicate themselves using this mDNA or maternal DNA. So when you hear about a study that talks about Eve, that talks about tracing populations back to a single maternal ancestor, the way we do that is through using maternal DNA or mitochondrial DNA. Mitochondria do some very important things. They allow ATP formation also by being important in the energy release from glucose that is broken down or used in the cytoplasm through the Krebs cycle. So ATP formation by this process called chemoosmosis is a major function of the mitochondrion. The mitochondria is also a membrane-bound organelle. It has two membranes, an external and an internal membrane, and the inner membrane is folded into structures called cristae, and it is on the surface of the cristae that ATP is generated. I am not going to go into great detail about some of these processes. I think that you may have done that as an undergraduate, talked to a great deal about the Krebs cycle and about chemoosmosis and how ADP is turned into ATP by the addition of a phosphate molecule as a way of storing energy, which is going to be then used as ATP is consumed by a number of cellular processes. The reason is that I want to focus on the parts of physiology that are actually clinically important, that are going to help you better understand clinical medicine and to prepare you for clinical medicine. So I don't think that all of the basic science has to be fully understood, but you have to understand that this process exists, that mitochondria exists, that ATP is important. And I will talk about those things as clinical concepts rather than having you doing math computations and so forth, or memorize things like the Krebs cycle. I just don't think that's very useful. So just to know, chemoosmosis is the process by which the mitochondrion makes ATP in its inner membrane. Protons are protons are transported using the electron transport system, and the energy from the flow of those protons is used to add a phosphate to ADP, making ATP. You don't have to remember that. But you do have to remember that ATP is essential for life. ATP does work. Among the most important work it does is pumps on membranes. So, for instance, when I talked at the beginning about that gradient where sodium is higher on the outside of the cell and lower on the inside of the cell, and potassium is higher on the inside of the cell and lower on the outside of the cell, that happens because there are pumps that use ATP as energy to move potassium in and sodium out. And we'll talk a lot about that in the next lecture. Again, do you need to know the details? Not the details, but you do need to understand that the sodium-potassium pump is an important structure for maintaining the electrochemical gradients that allow life to occur. ATP is used in synthesis, so ATP is the energy that is used when the body is synthesizing, when a cell, for instance, is synthesizing protein. ATP is also used in mechanical work. So muscle fibers, which use specialized proteins called actin and myosin to contract and do physical work, ATP provides the energy for that contraction. In addition, ATP provides the energy for cilia in our respiratory and gastrointestinal cells, where they're very important for function, and the amoeboid motion of white cells, which granulocytes, which have this ability to move in order to go to the sites of infection and to engulf bacteria, for instance, in order for the lysosomes to break them down. Cells have a skeleton, essentially, structures that are tubules and filaments that are composed of proteins. So some of the protein synthesis within the cell goes towards structure, towards this cytoskeleton, to support the membrane, to support specific structures on specialized cells that have cilia, to produce the centrioles and mitotic spindle that are important in cellular division for cells that are going to divide and reproduce. And in specific, again, specialized cells, actin and myosin are specialized proteins that contribute to contraction. There are also tubules, microtubules within the cell that can transport substances from one area of the cell to another. On to the next organelle, the nucleus. As you all know, the nucleus is where the genes reside, where the genetic material that contains all of the information that is needed for the cell to function exists. So the genes are in the nucleus. The genes control the production, the synthesis of everything really, but specifically of proteins. Both structural proteins, things like the cytoskeleton, and enzymes, and other proteins that are um, sometimes hormones that are produced within the cell. And the nucleus also controls mitosis, which is the process of cellular reproduction. Not all cells reproduce, and we will talk about this as we talk about some of the different areas of the um, different organ systems of the body. Cells that have specialized, for instance, into neurons no longer have the ability to reproduce. On the other hand, things like hepatocytes or liver cells are actively reproducing all the time. The hematopoietic stem cells within the bone marrow are constantly reproducing and um, specializing into specialized cell lines that reproduce up to a point. But once a cell becomes, for instance, a red blood cell, it is no longer able to reproduce. The nuclear membrane is also called the nuclear envelope. And here we have a little difference from the cell membrane. It is actually two membranes. So in addition to those membranes being lipid bilayers, which they are. They're the same lipid bilayer as the cell membrane. Here in the nucleus, we actually have two of those lipid bilayers forming the nuclear envelope. The outer membrane is continuous with the membrane of the endoplasmic reticulum. So there's a space between those two membranes, the inner and outer nuclear membranes, that is also continuous with the space or the compartment within the endoplasmic reticulum. There are pores on both layers, and various things diffuse through those pores. So here we have a diagram where we see two lipid bilayers. Okay. The inner and outer, the inner and outer membranes of the nuclear envelope. We have pores that are formed of proteins and other substances through those two membranes. And we have the outer membrane of the nuclear envelope continuous with the membrane of the endoplasmic reticulum. In here we have ribosomes that are both floating freely and ribosomes that are attached to the endoplasmic reticulum. Okay, so let's move on. Sorry, this thing is not. There we go. An exception to that idea that organelles have membranes is the nucleolus. The nucleolus has no membrane. It is within the nucleus, and it contains lots of RNA and ribosomal proteins. When a cell is actively synthesizing protein, the nucleolus gets larger. Also, RNA that is used in ribosomes is stored in the nucleolus, transported through the nuclear pores to the cytoplasm where it becomes mature ribosomes. Okay, so to build cellular structures, we have the rough and smooth ER, we have transport vesicles, we have the Golgi apparatus, we have the secretory vesicles and lysosomes. This is just a review. The rough ER has lots of ribosomes. That's what makes it look rough. Ribosomes synthesize the proteins, which are extruded into the compartment of the ER. Then the proteins may be within that compartment attached to carbohydrates to become glycoproteins. They may also be processed before they get to the Golgi apparatus by cross-links and folding. And then as they are pinched off into vesicles that are transported to the Golgi apparatus, they are further processed by things like cross-linking and folding. Within the smooth ER, the lipids like phospholipids and cholesterol are synthesized, and then they're incorporated into the bilayer. That lipid bilayer then can be fused with the membrane of the Golgi apparatus, from the Golgi apparatus to the cell membrane, fused with the cell membrane. So again, secretory vesicles are destined to go to usually one side of the cell where they fuse with the cell membrane and the contents are dispersed into the interstitial fluid. Lysosomes are used within the cell. Now we're going to talk a little bit about genetic structure and function. So genes control protein synthesis, and protein synthesis controls all functions in the cell. Proteins catalyze reactions, proteins give structure to the cell, proteins also are produced within the cell to be used outside of the cell for various reasons, and we'll talk a lot about this as the course goes on. The structure of the gene, and I know that this is review from undergraduate physiology, is that it is a double-stranded helix. So there is a double-stranded helix of DNA, deoxyribonucleic acid, and DNA is used to synthesize RNA, or ribonucleic acid, and then RNA functions to produce proteins. So the backbone of DNA is phosphoric acid and deoxyribose, and that backbone then fuses with bases, and there are 4 bases in DNA. 2 of them are purines and 2 of them are pyrimidines. Okay, so adenine and guanine are the purines, and cytosine and thymine are the pyrimidines. And what's called a nucleotide consists of 1 molecule of phosphoric acid, 1 molecule of deoxyribose, which is a sugar, and then 1 of the 4 bases. So there are 4 possible nucleotides. Okay, and they the bases, as I said, are attached to the deoxyribose, and then as the 2 helixes wind together, there are loose hydrogen bonds between a purine base and a pyrimidine base attaching to each other, and this allows the strand to be pulled apart when RNA synthesis is happening, and then to rejoin. So a purine always bonds with a pyrimidine, and more specifically, adenine always bonds with thymine, A and T, and guanine always bonds with cytosine, G and C. So you have these bases, right? And when there are 3 bases, that is called a code word. And the genetic code is formed from this stringing together of successive code words within the DNA that each code word is associated with an amino acid. So code words strung together, 3 bases at a time, create the code for the production of RNA, which then codes for the amino acids which will be assembled into proteins. There are code words that aren't associated with amino acids. So be aware that there's a chain initiating code word. It says this is we're about to start forming an RNA to code for a protein. There are 3 different chain terminating code words, and then there are 60 code words that correspond to the 20 amino acids. Okay, so this allows us, with 100,000 genes, to have a huge number of cellular proteins. In general, each strand of DNA carries the code for about 4,000 genes. So within the nucleus, where the DNA exists, RNA is manufactured. And that process of taking the genetic code on DNA and manufacturing RNA is called transcription. Once the RNA is manufactured, it goes through the nuclear pores into the mostly into the endoplasmic reticulum. So there's an enzyme, and remember, enzymes are proteins, right? So this enzyme has been produced by this very process. There's an enzyme called RNA polymerase that separates the DNA strands. One of those strands is used as the template. Those code triplets or words on the DNA cause the formation of complementary code triplets in the RNA. So RNA is a little different from DNA. The backbone is ribose instead of deoxyribose, and instead of thymine, uracil is the pyrimidine that is associated with cytosine. So it's very similar in a lot of ways to DNA, but there are some differences. One of the main differences is that while DNA is a double strand, it's a single strand of the DNA that functions as a template for the RNA, and the RNA, when it is formed and breaks away from the DNA, is a single strand, not a double strand. Let's see. So this is a place where ATP is extremely important. The phosphates from ATP are used to store energy to produce to synthesize the RNA, to promote the reactions that are important for adding the next nucleotide to the RNA chain as the process is continuing creating RNA from DNA. The bit of DNA that's ahead of the gene that's going to be transcribed is called the promoter. So the RNA polymerase finds the promoter, attaches to it, separates 2 strands, unwinds, and separates this helix. And then that polymerase moves along the DNA and forms RNA molecules by finding a complementary nucleotide and adding it to the DNA. And so here you see the unwound DNA, so TACG, right? And this is the strand that's not being used, and this is the template, and then the RNA polymerase is going to move along and add uracil to adenine, cytosine to guanine, and so forth, making a new strand of RNA. When the RNA polymerase gets to the end of the gene, it finds another sequence of DNA called the chain terminating sequence, and it breaks away, releasing the RNA. These are just some kind of terms that people use. If you think about it, the RNA polymerase is kind of going along and doing the work. The ribosomal RNA, it becomes the ribosome where work is going to happen. There is messenger RNA that takes the genetic code to the cytoplasm to control the formation of proteins. That's sort of called the blueprint. And then the transfer RNA is another kind of RNA that brings amino acids to the ribosomes to be used in the assembly of proteins, and that's the fetch and carry team. So there are 20 different types of transfer RNA. There's one for each of the 20 amino acids. And one end recognizes the codon, the 3-letter word, and the other lines up the amino acids. Let's see if we have a picture of that. Here we see the transfer RNAs out here grabbing onto their respective amino acids. We have our messenger RNA here, and those transfer DNAs are lining up with the codons in the ribosome and bringing them to make this strand of protein. The code on the transfer RNA that allows it to recognize a codon is called an anticodon. Here again, this may be too much detail. So translation of RNA results in the production of proteins. And again, this picture. Okay. Nuclear DNA is packaged in chromosomes. You know about chromosomes. The DNA is wound around proteins that are called histones. When it's wrapped, it doesn't function to form RNA. It requires unwrapping within the nucleus to make RNA. As you might imagine, only certain areas in the genetic code are unwound and active at any given time, and much of that code is packaged in these wound up structures called chromosomes. So let's talk a little bit about the process of cellular division or reproduction called mitosis. Some cells in the body have very short life expectancies. Cells in the bone marrow are reproducing constantly. And any given cell has about a 10-hour lifespan, so they have to continue to reproduce. On the other hand, cells within the nervous system, particularly the central nervous system, are formed in infancy and childhood and last a fetal life, infancy and childhood, and last a lifetime. So some cells never divide. Neurons never divide, muscle cells never divide, mature red cells never divide. In order for mitosis to occur, a replica of the DNA has to be made. Okay, so there's one duplication turning each double helix of DNA into two double helixes of DNA, two exact replicates. This is and unlike RNA, it's not a single strand. You're replicating both strands. And it's not just a part of the DNA molecule, it's the entire molecule. So it's a different enzyme that's used, and that enzyme is called DNA polymerase. The newly formed strand of DNA attaches by loose hydrogen bonds to the original strand that was used as a template. So in this case, each strand of the DNA is being used as a template, forming again from one helix two helixes. Those two helices are sort of bound together, but they have enzymes that uncoil by cutting the helix, rotating it, and then resplicing it. So you have, instead of this two double helixes, two helixes bound together, you end up with two separate double helixes. There are repair mechanisms and proofreading mechanisms. So there are enzymes that go along looking for mistakes, finding the mistakes, cutting them out, and fixing them, so that there are very few errors in this process of transcription. If you remember, the process of making RNA from a DNA template was called translation. This is called transcription. If an error occurs, that's a mutation. So that's a mistake, it's a mutation, it's a change. And ultimately, it might result in making an abnormal protein, or it might result in the inability to make a protein, and that might have an effect on future cell function. For instance, if it is a protein involved in the control of cellular division, a mutation might result in uncontrolled cellular division, which would be what? Cancer. Or it might result in the inability to create some cell division. All right. So those two chromosomes, that now we have the duplicate chromosomes, remain attached to each other in the center at a point called the centromere. The duplicated but still attached chromosomes are called chromatids. And then structures that are located near the pole of the nucleus, called centrioles, start to move apart by polymerization of microtubules. So the centrioles, we talked earlier about protein microtubules. They grow apart, pulling the centrioles apart. And you don't need to know these details, but this apparatus eventually pulls apart the respective duplicate DNA strands, causing mitosis. And I'm going to skip by the phases of mitosis. The chromosomes kind of, as they're pulled apart, they condense back into easily seen, well-defined chromosomes. There are these kind of phases that occur, and ultimately the cell divides with two separate nuclei, two sets of chromosomes. So some cells, some tissues, degenerate if there's regenerate if there's not enough tissue mass, and that's particularly true of the liver. Again, there are some cells that never reproduce. There are some other rapidly reproducing tissues in addition to the bone marrow and potentially the liver, and they include skin, which is constantly, the basal layer of skin is constantly reproducing. The mucous membrane surfaces of your respiratory and gastrointestinal tracts are constantly regenerating. There are a lot of ways that growth is controlled. So there are growth factors, and the growth factors may be produced in surrounding nearby tissues, or they may be hormones that are coming from distant organs or tissues and being brought through the bloodstream into the extracellular fluid. Sometimes simply the space available controls reproduction. And then there's sort of some negative feedback. So the secretions, the substances that are made by a cell, may slow its reproduction if they are not taken away. So cells that are grown in tissue culture will often stop growing, while cells in a living system often have ways for those substances to be diffused away and not to limit growth. Once cells have differentiated to a certain point, specialized to a certain point, they'll no longer necessarily be able to reproduce with omnipotential into different genetic characteristics. And that happens because there's this constant processes within which genes are activated and inactivated during the phases of cell division. So the point of stem cell research is to look at omnipotential cells, cells that still have the ability to differentiate, and to grow tissues from those stem cells by using appropriate growth factors and other factors. And finally, I just want to talk a little bit about this concept of apoptosis. That's a word I don't know if you've heard, but that you need to know about, because many cells have their own death programmed within them. It is important that cells, since we're having constant reproduction of some cell types, that we also have death of cells as they become less effective at doing their jobs. So this term apoptosis means programmed cell death. It's a cascade of proteolysis, so from lysosomes, that disassembles the cytoskeleton. It allows the cell membrane to begin to disintegrate so that other cells can phagocytose the dead cell and basically reprocess its substances for use in other cells. So apoptosis is initiated by an enzyme, a protease, that's called caspases. And in healthy adults, there is this very fine balance of cell reproduction and cell death. It appears that in some disorders, and particularly neurodegenerative disorders, that abnormalities of apoptosis are part of the uh, abnormality that creates the disease, the um, abnormal physiology. We certainly know that's true in cancer, that in some cases the loss of the cell's ability to die with programmed cell death becomes the focus of uncontrolled growth. And it also seems to be true in some autoimmune disorders. And understanding this process has allowed for the development of some drugs to try to help induce apoptosis in these abnormally reproducing cancer cells.